Yay, there we go. Oh, now. There we go. Okay, so this is the walkthrough for A-Level Biology Component 1, Energy for Life. The second paper, the A2 paper. So this looks fairly like what you're going to get in the exam, because I've just copied the SAMS stuff across. So you will need to remember in the exam to put your name and the centre number and your candidate number. This is the grid for the examiners, so this tells you what your questions are out of. So if you sort of prefer to do the longer questions first, why not look to see what they're going to be. It tells you that in addition to this, you'll need a calculator and a ruler. Calculator, ruler, we should have it in all exams. Use black ink or black ballpoint pen, do not use gel pen. That's because gel pens don't scan in very well and these are going to be marked online. You must not use correction fluid either, so you can leave that at home. You need to answer all of the questions, write your answers in the spaces provided, and then you've got the number of marks is given at the end of each question or part question. So you know that you're aiming to answer 9 questions, that's what the marks are out of. If in the real exam you get to sort of, you've got no room to write your full answer, you must put at the end, when you get to sort of this bit of the line, put extra sheets and turn to the end and there'll be a number of lined extra sheets for you to write that response on. Now those get scanned in then and attached to your answer. So, question 1. Oh, and I see a diagram of ATP and I recognise that instantaneously, and so should you. So it tells us that the diagram below represents a specific molecule which is used in many processes within an organism. A. Identify the molecule. So this molecule is ATP, and I'm going to put the full name because sometimes if they want the name, they say name or identify, that's what they really want. So this is adenine because it's got 3, phosphate. And it should be adenosine. See, I've just done exactly what you lot did in the exam, where a lot of you went, adenine, and then you got it wrong, aden. I can't even spell adenosine, I don't know why I'm trying. Adenosine triphosphate. And I'm going to put brackets ATP there. So, name the parts of the molecule by labelling the diagram above. This is what we call a rubric. That means you've got to follow the instructions. So no use just writing them anywhere, you've been given lines. A, that's the phosphates, the 3 phosphates. C is the sugar. I'm going to name that. I know it's a pentose, and if in the exam, if you're thinking, it's a pentose but I can't remember what its name is, write pentose down, might be worth a mark. And then my base, now this one really is adenine. And again, when I was marking this, I got a lot of adenosines for that. Adenosine is the base and the sugar linked together, but without the phosphate, so that would be adenosine. So you do have to be careful, like I wasn't. There we go. But that's what proofreading your work at the end of the question's about. So, give 2 examples, and they've given me the numbers 1 and 2. Processes which require large amounts, don't like that word, amounts of this molecule. So I want something that's going to use a lot of ATP. So my top 2 are always active transport because all cells do that. It doesn't specify what sort of cell, so I'm going to go for muscle contraction. But you could have had protein synthesis, that would have been good. And here I got sort of a lot of, used in respiration. No, not really. Respiration makes ATP. a lot of Calvin cycle, but remember that the Calvin cycle's only using the ATP that it makes, so it's not using like loads of it, so do be careful. Distinguish. Part C, distinguish between the terms oxidative. I'm going to put a box around oxidative. I'm going to write oxygen above it to remind me to talk about that. And phosphorylation. And then I've got photo, that means light. And then I've got the phosphorylation. Now phosphorylation reactions are technically just adding phosphate to a molecule. So, you know, activation of the glucose at the start of glycolysis is a phosphorylation reaction. But these two terms in particular refer to the electron transport chain making ATP, and remember we've got non-cyclic and cyclic, and they're churning out ATP. So phosphorylation in this particular context, and given that we've got a diagram of ATP, means making ATP. So oxidative phosphorylation, great word, phosphorylation. Phosphorylation is making ATP using oxygen, a final electron acceptor. So that's in the Christie, isn't it? And photophosphorylation. So you have to say something about each if you're distinguishing. Photophosphorylation is making ATP, that takes care of phosphorylation. Using light energy to excite electrons. So then obviously I proofread that, and I would have corrected my adenosine even without Dr. Saville's lovely help. Wouldn't it be great if you had Dr. Saville sitting next to you in the exam going, it's not that, it's not that.